Hi everybody, welcome to Biostats. I'm fine with you having your videos on it. I kind of like being able to see some people, but no requirement at all. I'm not gonna lie, I had my video off in a faculty meeting yesterday and fell asleep. So that's one reason to have your video on, because you're a little bit more accountable to actually being in class. It was real boring in my defense. Overall updates for Biostats. Thanks to everyone who did their reflection. If you did that at any point, I gave you full credit for it. I just really wanted to hear about it. If you're still missing it, try to get on and do it. It's just a Moodle assignment that I wanted you to think about what you're doing this semester. And most of you mentioned some worry about how to keep motivation up. I totally get that. Like, I worry about it too. It's really easy to be distracted at home and stuff. So we're gonna try to make a lot of things with due dates just so that you know that things are due. That said, if something's going on in your life, like you actually get sick or a family member's sick or something's going on with class, you just have to communicate with me, and we'll try to work it out. But I'm going to try to be really strict on deadlines so that people don't fall behind and get super late on work. In this class, we're pretty lucky that we're soon transitioning away from lecture and going into projects. So I really only had 2 more lectures that I wanted to give you guys. And then once we're doing projects, that's like pretty active. Like, you'd be working on it on your own. You don't have to worry about, can I sit and listen to my professor drone on? You're just going to be working through it. And I mentioned this, but to keep environmental studies a little bit more connected, or anybody can follow us, we started an Instagram account, Purchase ENV, so that we'll kind of be able to communicate that way as well. So hopefully, if everybody has found the raise your hand button, that's the way to get me to stop if you need me to, or just unmute yourself and talk. Easy way to unmute yourself is to hold down your space bar. And while it's held down, you can talk. You'll be unmuted during that time. So just stop me like you would in class if you have questions. What was that sound? Okay. So updates on the schedule for next week. So everything kind of was changing so fast that I waited. I think this is the best course of action. So today we're going to go over ANOVA, one of the last tests we needed to learn. And then on Monday, we're going to go over non-parametric methods, which is actually a pretty quick lecture, and then have some time to review together as a group. Thursday, again, I said this, I wanted to keep the quiz on Thursday. So quiz 3 is going to be take home, open notes. I'm going to say I discourage you from using the internet too heavily, because you will find things that aren't answered in the way that I want them to be answered. So I really want you to focus more on your notes and the book and how to interpret it the way I've been teaching it. It'll be pretty obvious to me if you Google all the answers and write answers from Wikipedia, because they talk about it in a totally more pure statistical way, and we've been talking about everything in a very biostats way. That's kind of how it's going to be. I modified the plan a little bit from last time now that they're kicking everyone out of the dorms. I don't expect the quiz to be longer than a regular class period, so that hour 40 minutes that you usually have for class. It's probably still going to be 25 questions, some similar to before, some that kind of build on questions, because now you're going to be able to look at your notes. I'll have more details on that on Monday. But I originally said it was going to be during class time. And now I just feel like that might not be the smartest way of going. So instead, I'm going to make it available at noon on Thursday, and then it's due Friday at midnight. So you have more than 24 hours to work on it. I just really think you could very easily get it done during our allotted class time. But if something's going on and you're moving out of the dorms or other things are happening, email me and we'll work something out. I just really wanted to get this quiz done before we go on spring break, because they've moved spring break up now. Partially, I think, to give us more time to get our classes situated. So I didn't want to have content spilling out through spring break that you had to remember. So we're going to keep that quiz on Thursday, but you have plenty of time to work on it. So it'll just be a Word document that you download from Moodle and put your answer your questions and resubmit it to me. Andre, you got a question?
I was just wondering, can you hear me?
Yeah.
Okay. I was just wondering, for the quiz, is it going to be like a Word document or is it going to be something that's like on Moodle? Like,
No, I'm just going to do a Word document
okay.
that then you can type into and resubmit it like you would a homework or something.
All
Um,
right.
so just an
Um,
assignment.
all right. And I wasn't here last lecture for when we went over like all the linear regression and all that.
Mhm.
But is there going to be any like for example, inserting any symbols like alpha or beta? Because that's one thing I have a little concern about for.
No, because you can just write it out. You can type it out, basically. Um, I'll make it all typable. I'll say that. Because if you do have to refer to alpha, you can just write out alpha instead of writing
Okay,
the
okay. I
symbol for
just, yeah.
alpha. Because that's like alpha is also significance level. So you could write it as significance level. You can write it as the word alpha, all of that. So no, it'll be like text friendly, like no drawings or anything, because I don't want to deal with how to integrate that at this point.
Yeah.
There might be more questions when we go to like project based stuff where I want you to like fill something out and take a picture and send it back, but I think we can work around that really and deal with that when the time comes.
All right. Sounds good.
Okay, thanks. Is there another question? Oh, Eric, you got a question?
Yeah, can you hear me?
Yeah.
So just wondering, is the problem set still going to be due that Thursday?
No, now it's just this instead.
So we're just not having that problem set?
Yeah, no. I think I'm going to work it into stuff after spring break, when we're doing more project-based stuff. Because basically now we're doing these tests. The more interesting thing is to integrate it with R anyway. Both of these tests, there isn't any math that you can do without using a computer. So we'll just do it. We'll do my problem set for ANOVA and regression when after spring break, basically.
Okay, and then the only other thing is, are we going to have a lecture on that Thursday, or is it just going to be the quiz for that day?
Just the quiz.
All right, thank you.
Because my hope is that most of you could just sit down at 12 when it goes live and just finish it before the class period would be over. That's kind of my goal. I lost my chat. Is anybody chatting? I don't know why it disappears on me all the time. Okay, no one's chatting. Okay, so moving on from that. A little bit more details. So how do you prepare for the take-home quiz? I posted a review sheet the same as before with questions and stuff about ANOVA and regression. So basically, once the quiz opens, Jermaine and I are not going to be answering any questions about biostats. So like I'll answer questions the way I answer questions during a quiz, which is like if you don't understand the question or just need clarifying on it. But I'm not going to be like, once the quiz is open, you can't come to me and be like, can you explain a p-value again? Because that's going to be a question on the quiz. So I encourage you then before Thursday to organize your notes and make sure you understand everything that we've gone over. If something's unclear, we're going to have time on during Monday class, and then I'll have my normal office hours. They're just going to be on Zoom. So I'll be in here and you can log in and ask me questions. I haven't talked to Jermaine yet about if he's going to do a review session, but he probably will if people are interested in it. I want you to think of this as not a high stress, you need to like cram study, but you do need to understand the material before you go into this quiz because it's going to be hard to kind of glean some of that information on your own. Does that make sense? Michelle, you got a question?
Hi, can you hear me?
Yeah, now I can.
How will Jermaine be doing the review session?
It'd be on Zoom too.
Okay, cool.
Yeah, we're not allowed to be on campus anymore, so
Okay.
everything's on Zoom.
Okay, thank you.
Yeah. Okay. Any other questions about that? So how I would organize my notes. Now I'm going to do something even fancier. Let's see if I can make this work. Can you guys see this now? Okay. So the way I would organize my notes if I were you guys is something like this. So we've gone over a bunch of tests. This whole quiz is going to be about the test, right? So there'll still be questions about confidence intervals and p-values and stuff because that relates to everything. But what I would do is go through and organize your notes. So like the first test that we learned, right, was a, I can't look at this, it's going to make me puke, but an unpaired t-test, right? Which is also called independent samples t-test, students t-test. Does anyone remember when an unpaired t-test is used, what type of data they have? I guess raise your hand. Ooh, biostats hurts your brain. Oh, Zach, go ahead. Got to unmute yourself. Can't hear you yet.
Separate populations. Can you hear me now?
Say that again.
2 separate populations.
Yeah, so use when you have 2 populations and want to compare means, right? So you would have those basically if you had population A and population B and you had a mean and a confidence interval, and population B, you had mean and confidence interval, are these 2 different from each other or not, right? So the null hypothesis for this is just that there is no difference between the populations. And the alternative is that there is a difference. The formula, you'd go to your formula sheet and write it out and make sure you understand it, right? The T, no, I didn't print out the formula sheet, but I'll make sure those are posted online. So like the T value, the T formula is T, and you need to make sure you know the mean of each group, the standard deviation of each group, and the sample size of each group. So making sure you kind of can understand that formula, identify the formula. What you need to calculate a p-value for this one, you need to determine the critical t-value. Does anyone remember how we get that critical t-value? Michelle, go ahead.
So I think you just use the chart and you find your degrees of freedom by doing n1 plus n2 minus 1. And then you use the percent confidence that you want, and then you use the chart to find the t-value.
Yep, so you do your confidence level and you get your t-value, right? The degrees of freedom was technically n1 plus n2 minus 2, and your confidence level. And then you get some sort of t critical value, like, and that's equal to something, and then you're going to calculate your t-value, which is equal to something. If your t-value is greater than your critical value, what do? Who knows that one? So if the t-value you calculate from this formula up here is greater than your critical value that you looked for, is this a reject or a fail to reject scenario? Zach, go ahead.
Reject.
Yeah, so this is a reject, right? This is that there is a difference. But hope this is all coming back to you guys. I know we haven't really done stats in a while. So I would fill all that in. I'd go in, some of them have like extra information that you're kind of going through. This would be something like all the assumptions, all that. So basically you could start making a page for each of the tests that we know, why we use it, what are the null and alternative, all that type of stuff. That's how I would start organize my notes, because that's going to be really handy when you go to take the quiz, that if I ask about an unpaired t-test, you go to your sheet of notes about that and you just know the answer right away. Any questions about that? Okay. That's my recommendation for study guiding. Oh gosh, I have to stop sharing. And now I'm going to share my PowerPoint again. Okay, so that's how I would organize my notes. So as you're in this class, I've kind of been thinking a lot about how to keep you guys motivated. So I would still take notes like you're in class if you can, because I think that keeps everyone kind of awake. Definitely raise your hand, ask a question to stop me. And I'll try to ask questions like I just did so that you guys can know that a question's coming at some point. I have some questions at the end of this PowerPoint that I'm going to ask that we can go over. And the same as I have during class, like I don't usually write the answers to the questions in my PowerPoint. So that's why you need to be taking notes as we go so that you have the answers in your notes. So what we know already, we're farther along in biostats than you would think just because of how I set up this class to structure everything kind of lecture heavy in the front, and then we'll go to project at the back end. So you already know the normal distribution, confidence intervals, hypothesis testing, what p-values mean, and then you know all of these tests, the unpaired t-test, the paired t-test, the chi-squared test, correlation and regression. And the last two things that we need to cover are ANOVA and non-parametric tests, and that's what we're doing now. So ANOVA, that's the kind of thing that's going on. I love some of these statistics memes. In the whole scheme of things, we're really so different. Like, yeah, we maybe you are. Like, maybe your means are different, and that's what ANOVA is going to kind of find out. So ANOVA stands for analysis of variance, and that's kind of, it's one of the tests that we build towards and kind of end in this class because it's something that's used by everybody. So you probably have seen this in a paper at different points, and then ANOVAs can build off of them to the point where you can get two-way ANOVAs and analysis of covariance and all these different things. But all you guys need to, we're going to kind of stop with ANOVA in this class. And if you go to grad school or continue on, there'll be more tests that kind of build off of ANOVAs. So the book chapters for today are chapter 39 and chapter 40. And then what I want to talk about, so we just went over the t-test, right? So the limitations of a t-test are that you can only compare two groups, right? That's how this works. So if you had a contaminated lake and an uncontaminated lake, we could compare fish lengths between contaminated and uncontaminated. But what if you had more than two groups? What if you were studying two different contaminated lakes and two different uncontaminated lakes, and you wanted to know if uncontaminated lake 1 was different than contaminated lake 1, and also was it different than contaminated lake 2? So now we just have more than two groups. You could go through and do a t-test between each of these. So like 1 to 1, you know, like go through and kind of do all pairs of t-tests, but that is really bad statistical design. So you see people do that every once in a while, and it makes me crazy because we have something called an ANOVA that can deal with this a lot better and more robustly. So what you're going to want to do if you have more than two groups is use an ANOVA, this analysis of variance, and that's what we're going to go over today. So the lowest level of ANOVA is called a one-way ANOVA. This compares the means of 3 or more groups. So t-test is stuck with only 2 means. ANOVA can do 3 or more. It's called a one-way because you're only comparing one treatment, but that means the treatment can have multiple levels. So for our lake example, the treatment is like basically site contamination level, but then our different treatments are contaminated lake 1, contaminated lake 2, uncontaminated lake 1, uncontaminated lake 2. So the treatment has a whole bunch of levels within it, but we can only look at this one treatment that we're talking about. So you can think of a treatment as a category or a type. It doesn't really mean that there's some sort of experimental intervention. Treatment can just be a difference between sites. It can be a difference between species. It can be a difference between habitats. Any of those things kind of fall into this thing that we call the treatment. So don't think of it as like I need to be doing something to all of these groups. It's just basically the 4 or the 3 populations that you want to compare, however you group them, those are your treatments. So interestingly enough, if you did an ANOVA with just 2 groups, it would give you the same p-value as a t-test. So it runs a very similar way when you only have 2 groups, but it is now able to deal with having a lot more groups that you want to get means for. Does that make sense to everybody? Pause for a second, raise your hand if you have a question. Okay. I mean, sometimes you guys look at me like, okay, Eric has a question. Eric, go ahead.
Yeah, so would it give you the same value as like either type of t-test? Like if you were using if you were comparing just 2 separate populations, it would give you that value. But if it was like one population that had a treatment on it, would
Yeah.
it?
No, we're going to go into, so like there is a different type of ANOVA that's similar to a paired t-test, and that would be the same. It's at the end of this PowerPoint, it's called repeated measures ANOVA. So that's the same as a paired t-test. This is the same as an unpaired independent samples t-test.
Okay, thank you.
Mm-hmm. Who's next? Andre?
Oh, I was just wondering. So basically, like for example, when we did our ecology project last semester, like would be comparing the means of those. Like it was 4 different plant groups using.
Mm-hmm.
Like that's like an example you could apply to ANOVA.
Yeah, because you just want to know, is one of the means of this different? Did one group grow differently than the other, basically? But I want to look at all my groups at one time. I don't want to do this group versus this group and then this group versus this group, which is what you'd be stuck doing if you only knew the t-test.
Okay, gotcha. All right, I just wanted to clarify.
Yeah, no, that's a good example. Any other questions? Sorry, I do have a hard time whenever I put the PowerPoint on presenter mode, I can't see the chat anymore today. So I see that Kim answered Sammy's question, and I thank you for that. It's going to disappear for me. So if you guys see someone chatting because they can't hear or something's going on, just raise your hand and stop me, because the raise your hand thing pops up, but for some reason the chat gets hidden. So just help each other out if somebody's having trouble. So that's the one-way ANOVA. So the assumptions of the ANOVA are the same as the t-test. You need to have randomly selected or representative of the population samples. All of your observations have to be independent. The data should be sampled from populations with a Gaussian or a normal distribution, and the standard deviations of all the populations are similar. So again, this one is fairly robust to deviation from normal, but we're going to talk more about, like, we need to have generally normally distributed data to work with this. So the null hypothesis in a one-way ANOVA is basically that all populations share the same mean. There's no difference in means between the populations. The alternative hypothesis is that all populations do not share the same means. What this means is that at least one of your populations has a mean different from the rest. It doesn't mean that every single one is different from each other, which is kind of annoying, and we're going to get into how we deal with that. It just says that at least one population is different. So it could be that all of your lakes or all of your treatments have totally different means, but we don't know that based on the results of your one-way ANOVA. All we know is that there is one mean that's different than the others. And we're going to run another test to figure out which one's different. So the p-value, and this one, a low p-value means you're rejecting the null hypothesis. It means that at least one population has a mean different than the rest, right? So this is why we spent so long on p-values in the beginning, because basically, like, they just keep coming up. Whenever you have a low one, you're rejecting your null. High p-value, you fail to reject your null. There's no difference among the means that you're studying. Any questions? Okay, so how do you calculate it? What you're going to do is the same way we got like a t-value or a chi-square value. You're going to try to get an F statistic. That's the thing that's tied to ANOVA. A large F value means a low p-value. So the same method as a chi-square or a t-test, you're going to compare your F statistic to some critical value. If you have an F bigger than a critical value, you reject your null hypothesis. If you have a large F and a low p-value, that means at least one group is different. it uses the same principles of sum of squares that we talked about in linear regression, but it's a little bit different in ANOVA. So I want to show you this video first. Let's hope this works. Can everyone see YouTube now? Cool. Okay. So let's watch this together. They do a really good job, and I didn't want to recreate the wheel. Can you guys hear that too? Did you hear the YouTube video playing? No. Okay, hang on. Can you hear that now? Huh. Hold on. Gotcha.
Now it is time to begin the one-way ANOVA.
Do you hear it
A
now?
one-way ANOVA, remember, has one factor with at least two independent levels. So here's an example of that. Researchers want to test a new anti-anxiety medication. They split participants into 3 conditions, 0 milligrams, 50 milligrams, and 100 milligrams, then asked them to rate their anxiety on a scale of 1 to 10. Are there any differences between the 3 conditions using an alpha of 0.05? So here are the 3 conditions. There are 7 people in each condition, and there are their scores. So we're going to do an ANOVA using these 7 steps. And I've already gone over this a few times, so I'm just going to go right through it. First is stating the null and alternative hypotheses. Now in an ANOVA, our null is that the 3 groups are equal to one another, and the alternative is that not all the groups are equal. That's it. Our alpha level is what I said it would be, 0.05. Just use whatever is stated, 0.01, 0.05, whatever. In this case, we're using 0.05. Next, we're going to calculate the degrees of freedom, and we're going to need 3 of those. We're going to need degrees of freedom between, degrees of freedom within, and degrees of freedom total. Now, before I do that, we need to make sure you understand the distinction between capital N and lowercase n. Lowercase n in this case is 7. It refers to how many people are in each level of the independent variable. And capital N refers to the total number of people in the experiment. So lowercase n is 7, capital N is 21. We're going to use that to calculate the degrees of freedom and some other things. So these are the equations for the degrees of freedom. You see we have n, we have a, we have 1. A refers to how many levels of the factor that you have. So in this case, we have 3 levels, 0, 50, and 100. So A is 3. So that makes our degrees of freedom between 2, our degrees of freedom within 18, and degrees of freedom total 20. Notice that total is just between plus within. 2 plus 18 equals 20. If between plus within don't equal total, that means something went wrong. Next, we will state our decision rule. To look up the critical value we need to use 2 different degrees of freedom. We're going to use degrees of freedom between and degrees of freedom within. We're going to use 2 and 18. Now we're going to use this F table, which looks very complicated. Hopefully you're watching this in high definition so you can actually read what these numbers are. This is the smallest F table I could get into this lecture. But this is the F table. On the top we have the between groups degrees of freedom, and on the left side we have the within groups degrees of freedom. So I'm going to use between is 2 and within is 18. And that's how I'm going to find the critical value of 3.5546. So our decision rule is, if f is greater than 3.5546, we're going to reject the null hypothesis. Next, we need to actually calculate the f. So this is an F table and ANOVA printout table, and what we're going to do is we're going to use this table to find F, the area that I have indicated in red. The whole thing we're doing right now, the whole point of this is to calculate a single F value, which we're going to compare to the critical value. So I'm going to put in the degrees of freedom that we already know, 2, 18, and 20. And next, we're going to calculate the sum of squares. We're going to need sum of squares between, sum of squares within, and sum of squares total. This is the equation for sum of squares between. Now, most of it makes sense, except for that weird sum of AI squared thing. Really what that means is you're going to take the sum of each level, square it, and then add those together. It'd be easier for me to show you, which I'm going to do. You're going to take the sum of each of the three groups. You're going to get 57, 47, 21. You're going to square each of those sums, and then you're going to add together all of those sums. That's what that means. So hopefully if you're good with that, you should be good with this whole equation. Now, t squared, well, t just refers to the total sum. So if you add all the numbers together, if you add together 57, 47, and 21, you get a t of 125. And we already know lowercase n and capital N. We know that they're 7 and 21. So we can find sum of squares between. If we solve for it, we get 98.67. Next, let's do sum of squares within. Sum of squares within is sum of all y squared minus, hey, this thing, we already found it when we were calculating sum of squares between. We already know the second half of this. We already have that, so we can just plug that right in. We just need to find sum of all y squared. What that means is you have to square every individual value and then add them all together. So our sum of y squared is 853. That's the first part of this equation. So now solving for sum of squares within, we get 10.29. And lastly, we have sum of squares total. Now the nice thing about this is both parts of it, sum of y squared and t squared divided by n, we already found both of those. We can just put in what we found before. And we find a sum of squares total of 108.95. So let me go back to this ANOVA table. I'm going to put in our three sum of squares. Now we're actually almost done. We're very close to finding the f. Now the f, in order to find it, it's just ms between divided by ms within. That means mean square. So mean square between divided by mean square within. We just have to find those two things. If we have those, we have f. Now these are the equations for mean square between and mean square within. All you have to do is take the sum of squares and divide it by the degrees of freedom. So like to find mean square between, you just take 98.67 and divide by 2 and get 49.34. Same thing within, you just take 10.29 divided by 18 and get 0.57. And now that you have both of those mean squares, you can just divide them to find an f. And we get an f of 86.56. Now everything we've done so far, it was just to get that f. And now we're going to go back to our decision rule, which was if f is greater than 3.5546, reject the null hypothesis. We found an f of 86.56, so that means we are going to reject the null hypothesis. Our conclusion in this case would be the three conditions differed significantly on anxiety level. And then the official way of writing that would be f218 equals 86.56, p less than 0.05. Now we just know that the groups are different somehow. We don't know where they're different. To actually figure that out, we would have to do a post hoc test. But from this one-way ANOVA, we did discover that there is a difference somewhere among these three groups. And that is a one-way ANOVA.
Okay. So now I'm going to go over my side of that. Let's see if we can get this back on. All right, you're seeing the PowerPoint again, right? Okay. So the things that he talked about in that video, right? So there's there's a whole bunch of different sum of squares. So the sum of squares between, this is also called the sum of squares among groups or sum of squares among. This is the sum of squares of each group mean from the grand mean weighted by sample size. Okay, so the grand mean is if you took a mean of all of the samples from all the populations, and now you're going to compare each group mean from the grand mean. Sum of squares within is the residual sum of squares or the error sum of squares, and this is the sum of squares of each value from its group mean. So how different is each individual value within Lake 1 from the mean of that group? And then the sum of squares total is just the sum of squares of each value from the grand mean. Or you can add up sum of squares between and sum of squares within, and that's your sum of squares total. So those three things are important, and he showed it kind of in this table form, which is how ANOVAs are always shown if you're trying to do them out by hand. So between samples is sum of squares between, within samples is sum of squares within, and then there's different ways of calculating the degrees of freedom for each. So he used A, I think. We're going to use K as our number of groups. So in our lake example, there were 4 lakes. So there was K number of lakes, or K equals 4. The overall sample size is all of the samples that you have in there. And so you just calculate the degrees of freedom in different ways, either K minus 1, N minus K, or N minus 1. And then you kind of move across this table from so you're kind of going to go across the table like this, right? And so now you've calculated the sum of squares between, you calculated the degrees of freedom, and then you're going to do the mean squares between, which is just taking this and dividing it by this. And that's what you get for your mean squares. You go across, you do that here, and then you basically have these 2 values. And then those 2 values, oh, I can't. So the K minus 1, this is basically showing that this is from this column here. K minus 1 is the degrees of freedom between divided by the sum of squares between. And then you move over and you get your F statistic, which is basically mean squares between divided by mean squares within. And that's how you get your F value in the end. Okay. Does that make sense? Was there a hand? No. Okay. So the important thing, remember when we did the t value, we talked about how the ratio here was the signal to noise. It's kind of the same thing for the F ratio. So the numerator is the mean squares between, how different the groups are from the other groups. And the denominator is the mean squares within, so how different each individual group is. So you want the mean square differences between groups to be bigger than the mean square differences within groups, right? You want there to be more difference between your different lakes than there is within a lake, because that's showing that there is a difference statistically between the lakes. So I hope this makes you happy. We won't be calculating ANOVA by hand, because most of the time when you're doing an ANOVA, you're using bigger sample sizes than he had in his example there, right? So we want to be working with maybe 30 samples per lake, and that makes it kind of unwieldy to do by hand. So you do need to know what this F ratio is coming from. So in general, that is the mean squares between divided by the mean squares within, how you get mean squares between and mean squares within, but you're not doing it out by hand, because that's what we're going to use R to do after spring break. Okay, any questions about that in general? Okay, so that is comparing groups that are separate, and we can do something called a repeated measures ANOVA. So NOVA is to repeated measures ANOVA as t-test is to paired t-test. So it's the same general idea, but you would use a repeated measures ANOVA if you made measurements repeatedly on the same subject. So like basically we talked about paired t-test or like before after. This is maybe you have a whole bunch of afters, so you have like time 1, time 2, time 3 on the same subject over and over again, then you would do a repeated measures ANOVA. There's also ways in like psychology or medical studies that maybe you recruited the subject as 3 matched pairs, studying triplets, you know, if you do something like that. But a lot of times in biology, it's that you got samples from the same individual. If you sampled multiple feathers from the same bird, they're going to be related to each other. And so you're going to do a repeated measures ANOVA. So moving on, what does an ANOVA actually tell us? So this is from a paper that I wrote where I'm looking at the mercury level of a whole bunch of different bird species in the Atlantic Highlands. So an ANOVA, if I did it on this data, I have an ANOVA and I got a p-value of 0.02. So if I had set my confidence level to 95% significance, what am I going to do in this scenario? What's my conclusion?
Reject or null.
Reject the null. Good. So we're going to reject the null. And what that means is just in a ANOVA, it means at least one mean is different. But I want to know, is this white-throated sparrow that's on the left side different than the rusty blackbird, which is on the right side, or is it different than the ovenbird that's right next to it? It doesn't tell me that. It tells me that one of these means is different than the other. Andre, you got a question?
So just to clarify from watching the video, so you reject the null if P is less than 3, what was it, 3.6?
Well, no, so that's using a critical value of the F statistic. So it's the same
Okay.
thing as what we did before. So you calculate this F statistic, and if it's bigger than the critical value that you got from the table, you're going to reject your null hypothesis. But if it's bigger than the critical value, that also means your p-value is going to be low, less than whatever you're saying.
Gotcha. Okay.
So they're related the same way as it was for chi-square or for
Yeah. All right. Thank
yeah,
you.
so if we got a p less than 0.02, we know that at least one of these means is different, but we don't know which one. That's kind of annoying, right? Like, I don't really care that at least one of these birds is different. I want to know which one it is, right? Because that's going to be the interesting part to me. So what we really want to find out is this. So like, you've probably seen these on graphs before where there's letters that signify which means are different from each other. Have you guys seen this before in general? So like in here, I have like letters under each of these error bars denoting if it's different from each other. So in the top left, estuarine and freshwater, the first one is AB, the second one is B. That means that there's B and B, they aren't significantly different from each other. But freshwater and upland, they don't share the same letters, so they are significantly different from each other. That's what we want to get to in the end, because we want to know where the difference is, right? So what we're going to do is, after you do an ANOVA, if you get a significant p-value, you're going to do a multiple comparisons post hoc test. So ANOVA fixes the problem of doing a bunch of t-tests, but it doesn't tell you which group is different overall. So you need to find a way to test those multiple comparisons correctly, right? So you don't want to just go through and do a ton of different t-tests, but you do want to know which one of these pairs is different. So what we do when we do these post hoc tests is that if you have that 95% confidence level, we're going to apply the 95% confidence level to all of the family of comparisons, and I'll get to what that means. So splitting the confidence level among comparisons means that they have to share the overall significance level, right? So they're sharing it among the family. If you're eating by yourself, you get to have all of the food. You get all of that alpha equals 0.05 to yourself. If you're sharing it among your family, you have to split it up, and you get, 1/4 as much food or 1/5 as much food. So the more comparisons that you make, the more times you're trying to see if things are different, the more you're going to split up your significance level between different groups. So we sometimes call this the family-wise error rate. It basically means that all of those pairs within your ANOVA need to learn how to share. Instead of each test getting 0.05 as their alpha level, you're going to share it among all of your tests. So now the 0.05 applies to the entire family of comparisons. And it results in a really conservative estimate instead of potentially creating more problems because you manipulated your data in a certain way. So the test that we usually use is a Tukey's test. This is called a Tukey honestly significant difference test, or the Tukey HSD. What it's going to do is do all that work of sharing the confidence level among the comparisons. And so what it's going to give you is, you ran an ANOVA with all of these different groups, and you know that one mean is different. Now it's going to do pairwise comparisons. So group A to group B, group A to group C, group B to group C, and say where your difference is, which means are different. So we look at this and we want the 95% confidence interval of differences to not overlap 0. So looking at this hypothetical data below, which one of these confidence intervals does not overlap 0?
Non-runner and versus recreational.
Yeah. So the bottom one, right? So we're looking at the differences and the non-runner versus recreational, the bottom one, that confidence interval does not overlap 0, which means that that pair has a significant difference between them, right? So these were different, running types. I forget exactly what they were studying, but in the first two, there's no significant difference between recreational versus elite runners or non-runners versus elite runners. But there was a significant difference between non-runners and recreational runners. So what do you then do is you conclude yes or no for each pair of comparisons. So this would look like no, no, yes. Those are where our differences lie, right? So now we've honed in on where the actual difference is between your groups. That's just another way of looking at that. So doing these multiple comparison tests, you're trying to reduce your chance of making a type 1 error. So remember, a type 1 error is rejecting a null hypothesis that's actually true. So it was our example that you tell a man that he's pregnant, right? There's no way that's true. So when you split up your alpha between all the tests that you're doing, you reduce your chance of making a type 1 error. The flip side of that is that it increases your chance of making a type 2 error. A type 2 error is failing to reject a null hypothesis that's actually false. So telling a very pregnant woman that she is not pregnant. So it's kind of two sides, right? So you want to, in general, minimize what types of type 1 error you make, but then be aware that you're going to increase your chance of type 2 error by doing this. So what do? We just can't do anything. Basically, you want to try to keep your treatment group number low. So you don't want to do an ANOVA with like 30 different groups that you're comparing because it's really hard to get accurate answers. Also, increasing sample size is always kind of the key to getting good results here. So the more treatment groups, the lower the statistical power to actually see differences. You can fight that by increasing your sample size within each group. Does the general idea of this post hoc test make sense? It's like, besides, I haven't really given you too much of the math behind it. It's mostly just that your ANOVA is just able to give you that one F statistic and the one p-value that says there is a difference between at least one of the means in this groups of means that we're looking at. And then you need to do this post hoc test to see where the difference is, where the mean is different. I'll take a breath so you can ask questions if you need to. Okay, so we talked about the Tukey HSD test, but there's also other types of multiple comparison tests. So Dunnett's test, you compare each mean with the mean of a control group. You need to decide this as part of your experimental design. I've never done something like this because I think it's more common in biomedical studies where you're actually doing experiments. But if you can set it up that way and work your design into it, it creates more narrow confidence intervals because you kind of are holding things more constant. Bonferroni's test, it can compare pre-selected pairs of means, but in general, Tukey is going to be better because it kind of applies to more things. Chef's tests, you can make elaborate comparisons, compare the mean of all treated groups to a specific group. It allows more ability to kind of generalize and make general comparisons, but it creates really wide confidence intervals. So in general, we kind of view Tukey's as like our catch-all. This is what works after our ANOVA if you're just working with normal data. If you had your design set up in a specific way, you would maybe choose one of these instead as your post hoc test. But I wanted to make sure you knew that they existed because you do see them in literature sometimes. And they're basically doing similar things to Tukey's, but hopefully the author that you read has a reason why they chose to do this one over Tukey's, because Tukey's is kind of the accepted one for post hoc tests after ANOVA. So the consequences of correcting for multiple comparisons: you have a lower risk of type 1 error, but you have a higher risk of type 2 error. This is all because you're increasing the width of your confidence intervals, which makes it harder to find significant differences. One of the curious things about this is that if a one-way ANOVA reports a p-value less than 0.05, are multiple comparison tests sure to find a difference between groups? Usually, yes. So usually if you get that high F statistic and the low p-value from your ANOVA, you're going to find one of your means is different than the others. But it's not necessarily true because of how we do this family-wise error in the post-hoc test. Because you're dividing up all your power and increasing the width of your confidence interval, there is a chance that all those pairwise tests will not be able to detect a difference, and you'll end up still not knowing which mean is different in the end. You'll have an indication and you'll have to figure out how to word that in the future. But it's not unheard of that you'd have an ANOVA that would say it was significant, and then you can't actually show where that difference is using your post hoc test. Okay, any questions? Okay, let's try to go over some questions on what we just covered. So the first one, when do you use an ANOVA? Can somebody answer that one? Michelle.
When you have when you're comparing 3 or more groups.
Yeah, so if you have just 2 groups, you would use a t-test. If you have more than 2 groups, you're going to use an ANOVA. Does anyone remember when you would use a repeated measures ANOVA then? No one? Michelle, is your hand raised for this? No, okay. So repeated measures ANOVA is similar to which t-test? Eric.
Yeah, so the repeated measures ANOVA is it's like a paired t-test. Basically you can use it if, like, say you run a treatment on some sort of group and then you measure it in a day, a month, and then 3 months. Like repeated measures ANOVA would work well there.
Yeah, so the same way if you had a paired t-test, because there's some relationship between your participants in the first and second group and you paired them, a repeated measures ANOVA just has a third pair, basically, right? So it's the same individual more than 2 times, you'd have to use a repeated measures ANOVA. Okay, so what are the null and alternative hypotheses in a one-way ANOVA? So let's start with a null hypothesis in the one-way ANOVA. Oh gosh, this is kind of fun actually. Kim, were you one of the people that raised your hand?
Yeah, you can hear me?
Yeah.
Okay, so the null for a one-way ANOVA is that there's no variation between the means of the however many populations you have.
Yeah, so it's the same thing as normal. There's no difference. Do you want to take what is the alternative hypothesis?
Sure. All populations do not share the same mean, so at least one of them has to be different.
Yeah, so you could put that both ways. The way I had in the PowerPoint was that they all do not share the same mean, but you could also say at least one population has a different mean, because that's the kind of better way to think of it, because of the fact that it's not telling you which mean is different. It's just saying that at least one is different between all of these groups that you sampled. Good. Any questions about that stuff? Okay. So what are the 3 sums of squares that are needed to calculate the F statistic?
I'm sorry, you said F?
The F statistic, yep. So remember, we get an F statistic to that's going to tell us if we are above this critical value for F. And so there were 3 different, there's 2 really important sum of squares, and then I talked about a third one. Kim, were you raising your hand for this one too?
Yeah. So one of them is sum of squares between. One of sum of squares within, and I don't know what the third one is.
Yeah, so sum of squares between, sum of squares within. So do you remember Alex, were you raising your hand too? Yeah, unmute yourself. What's up?
Because for some reason I can't raise my hand. For some
Oh, I
reason.
did see it at the last minute. You raising it, hitting.
Oh, okay, so it did work. Great.
Yeah. Mm-hmm.
So if I remember correctly, the sum of the squares between within and the total.
Yes, total's the third one. So total was kind of the bottom of the table, with between and within were what we were calculating as we moved across the table. So what do you actually use to calculate the F statistic, the F value that we're going to get? Anyone remember?
Still me or somebody else?
Do you know? Do you want to answer?
Give me a minute.
Okay, who else? Who else remembers the two that what we use, and one goes above the other? So do we want the mean squares between or the mean squares within to be bigger? Eric.
Within. Oh, sorry.
So if I remember correctly, it's like the mean of the sum of squares between divided by the mean of the sum of squares within.
Yeah, so you're moving across that table. Let me go back to that slide so we can all remember it. Where is it? So here's the slide, right? So we can get sum of squares between, sum of squares within, sum of squares total. You're going to calculate the degrees of freedom for each, and then you get something called the mean squared between, which is basically your sum of squares between divided by your degrees of freedom. And then the mean square within, which is the sum of squares within divided by your degrees of freedom for that. So then your F statistic in the end is your mean squared between divided by your mean squares within, right? So that means because we want a big F statistic or F value, you want your mean squares between to be much bigger than your mean squares within, which means you want there to be more of a difference between your groups than there is within each individual group. Does that make sense to everybody? So you kind of have to remember how all of this plays out. Obviously, for this quiz, you're going to be able to see this graph, this table in your notes, but you need to be able to say, like, yeah, if to get a difference, I want my F statistic to be big, and that means I want my mean squares between to be big, which means I want my sum of squares between to be big. Okay. So does a small F ratio correspond to a small or a large p-value? Alex?
Large p-value?
Yes, so a small f means you have a large p-value, and then the second part of that is what do you conclude if you have a large p-value? Somebody else can answer that one. Aya.
You fail to reject.
Yeah, so large p-value always is fail to reject. So now we got a small F ratio, we got a large p-value, we're going to fail to reject, which means there's no difference in the means between these groups. Good. So this is what I was just talking about. Is it better to have a high mean square between or a high mean square within? Ooh, Aya, you want to answer that one too?
Shouldn't it be that you should have a higher one between just as the numerator, and you want a bigger F?
Yeah, so you want a big F, which means you want that numerator to be bigger, which is that mean square between. And also just thinking about it theoretically, you want there to be a bigger difference between the different groups that you're looking at than within each individual group. So you want there to be a high mean square between. That makes sense to everybody? Okay, so what does the p-value in an ANOVA test tell you? Kim, go ahead.
I don't know if this is what this is exactly asking, but is it just the level at which you reject or fail to reject? I don't know.
Well,
I don't know how to be more specific with it, but like.
Yeah, so that's what you're using your p-value, right? So if your p-value is less than the alpha, you're going to reject. If it's higher than the alpha, you're going to fail to reject. So that's kind of how we use it functionally. But can anyone think of that old definition of a p-value that we used to talk about and how that relates to an ANOVA test? That old definition of a p-value. No one remembers it? Oh. Oh, Zach, you want to answer?
It's the probability that you would have the results that you have if there wasn't a difference between populations, or if the populations were the
Yeah.
same.
So our like standard definition of a p-value is the probability that you would see these results even if the null hypothesis was true. So you insert what we know about the null hypothesis here, which is that this is the probability that we would see these results given that there actually is no difference between the populations, or given that all of the means of all the populations are actually the same. Right? So remembering that like weird wording of a p-value is really important, and then we're just going to apply it to each of these different tests. Okay, so that's the end of ANOVA stuff. I think you should all take this little bit of time to go over it. I'll stay on the line until the end of class if you're looking over your notes and have more questions. So Monday, we're going to do non-parametric methods and then some sort of review so that we can all kind of get organized for the quiz. Wednesday is going to be lots of office hours for review if you have questions. Thursday, quiz 3 opens at noon and it's due Friday at midnight. If you need more time because you're in the middle of moving out or something, send me an email and we'll figure out how to deal with that. Happy to do it. But remember, once that quiz opens on Thursday, I'm not, for everyone, even if you're taking the quiz later, I'm not going to answer questions because you're going to be seeing the quiz in front of you. So once you want to try to get all your questions in before Thursday, if you're confused about something, so that then you can actually just look through your notes and get all the answers that you need. So thanks everybody. If you have questions, stick around. I can see the chat now, so if you want to type things into the chat to ask questions, feel free to do that. Otherwise, I'll see you guys on Monday.
Did you say whether you were posting the like lectures with your voice over it, or like our voices, or no?
Yeah, I'm recording this one and I can post that. I kind of also, I can't tell if everybody logged in today. It looks like a lot of people got to log in.
It said that there was just 21 people in here.
So. Okay, so that's close to everybody. So yeah, I'll post this recording basically. It'll be a YouTube link. It takes a little while to format it, but I'll have that.
Yeah, just like before the quiz, it'll be up.
Yeah, oh totally. It'll be up tonight at some point. I see Alexis's question. Yeah, everything's on Zoom for now on. So Zoom session Monday, where we'll have a little bit more lecture and some review, and then my office hours on Wednesday will be on Zoom. So I'll just be like sitting in a room like this, and you can log in and talk to me the way you would stop by my office. It'll just be virtual.
And you said that campus isn't advising teachers to come to campus anymore, or is
no it? Yeah.
Oh.
I know, it's a bummer, but what?
Interesting.
What was that, Anton?
That's interesting, just because of everything that's going on.
Yeah, I mean, they're trying, basically, like, if we don't, as professors, like, we don't have to be on campus now that we've got this all set up, so we're kind of starting to fall into non-essential personnel, and they've made a, like, anybody who can work at home should be working at home, just as every job should be doing. So they kind of are keeping us off campus. And I don't mind these Zoom sessions. They're easier one-on-one. So if people want help, you can still log in and pick a time to meet with me on my Calendly link, and we can talk that way.
Okay.
Yeah, and they're basically trying to get everybody off of campus. That's what Alexis wrote. Like,
Because there's rumors coming that people are getting confirmed cases on campus.
yeah, I've heard that too. I mean, it makes sense, like, that the cases are everywhere. So they are still doing the thing where you can apply to stay in the dorms if you need to.
And I think that they're like, I think there's almost like 800 or 900 campus cases that are actually in Westchester. So it's like, it's spreading very fast.
Yeah, so I think it's really smart for all of us. I mean, this feels like I'm talking to you guys. There's parts of the lecture that I hate, but like in general, it feels like we're still able to have communication. So I feel personally like I just have to stay home because it's not smart to go out and potentially infect other people. So I might run back to campus and get my computer monitor, and then I'm just gonna stay here for the rest of the semester, probably. Kind of sad.
18 months.
Yeah, I hope it's not that long. I can't handle that. I'll go crazy. I think everybody will. Kim, did you have another question?
No, I'm just hanging out.
Oh, okay.
Got nothing better to do.
Okay.
In quarantine.
Yeah. Oh, Andre, did you have a question?
Yeah, I heard you answer Anton's question about like this recorded lecture getting put up on Moodle. Will you also like post like just the PowerPoint?
Yeah, the PowerPoint's already up.
Cool.
So you should be able to download that and have it. Theoretically, if I have a ton of time, I'll voice over just the PowerPoint so that people who miss it don't have to listen to all of our technology, but that might be hard to do consistently. So I'm mostly planning on posting the Zoom thing.
Okay, I just want to make sure so I can print out like just the PowerPoint and.
Yeah.